Good afternoon. In the last video on the cell, we explained the structure and function of peroxisomes, but I am going back to the slide on peroxisomes because I would like to highlight some important pieces of information. The first thing I would like to highlight is that peroxisomes are one of the organelles that are surrounded by a single membrane. And as we said before, most of the organelles are essentially surrounded by one membrane or a single membrane. The ones that are surrounded by a double membrane, like the mitochondria and the chloroplasts, or the ones that are not surrounded by a double membrane, like ribosomes, are a minority. And therefore, you should every now and then return to the table that explains or that reminds you of the organelles that are surrounded by a single membrane, those that are surrounded by a double membrane, and those that are surrounded by no membrane at all. So again, this is, but when we say that it is surrounded by a single membrane, we mean that it is a single membrane like the cell membrane around the cell, which is a phospholipid bilayer made of two layers of phospholipids. The second important piece of information is that the peroxisomes have in the center a crystalloid core. And that crystalloid core is not always present, but when it is present, it is very striking and characteristic in electron micrographs when we use the electron microscope. And this center or crystalloid core, it contains enzymes. So you will find enzymes of peroxisomes accumulated here. And the enzymes of peroxisomes are usually oxidative enzymes, and this is very important. We usually think of oxidative enzymes as being harmful, but because they might cause oxidation in the cell. But sometimes oxidative enzymes are important, as we will see in the case of peroxisomes. What oxidative enzymes are present in the crystalloid core of peroxisomes? We have the catalase enzyme and the urate oxidase enzyme. The one that should be important to you, the one that you should remember, is the catalase enzyme. As we said in the previous video, that catalase helps the cell get rid of H2O2. So this is the second point I would like to highlight. And the third and last point I would like to highlight is that around the crystalloid core, we have a granular substance that looks more or less like the cytosol of the cell, but inside the organelle, and it is the site where biochemical molecules are present, whether the reactants or the products of the biochemical reactions. What is H2O2? On this slide, we're going to know more about H2O2. Under normal conditions and in non-cancerous cells, hydrogen peroxide is produced as a byproduct, as a side product in mitochondria. H2O2 is produced during cellular respiration in normal cells. And H2O2 is produced through special enzymes, but you don't have to know these enzymes. Even when we are going to study aerobic cellular respiration that occurs in the mitochondria, we wouldn't go over in detail, and you wouldn't be asked about these enzymes that result in the formation of H2O2 in the cell. Needless to say that H2O2 is an oxidizing agent, because if we leave H2O2 to dissociate on its own, H2O2 will dissociate into one molecule of water and one atom of oxygen. When we write the O with the brackets, those rectangular brackets, this means atomic oxygen. And atomic oxygen, of course, is a highly reactive oxygen, or oxygen in a highly reactive form. Why is it highly reactive? Because it is unstable. Oxygen should be in the molecular form in order to reach the octet state and to be stable and non-reactive. What are the sequelae or the consequences of having highly reactive oxygen? One important consequence is that this atomic oxygen will move freely in the cell and oxidize the organelles or molecules inside the cytosol and molecules inside the organelles, and result in the formation of harmful products of oxidation. And that's why we don't leave H2O2 to dissociate on its own, but we force H2O2 to be converted into two useful substances, water and oxygen. So this is not a spontaneous reaction. This is a catalyzed reaction. So when you leave it to dissociate on its own, it would give you highly reactive oxygen. And that's why if oxygen results from the spontaneous dissociation of H2O2, so that's why it would be logical to say that H2O2 is an oxidizing agent, okay? So what is the evidence, or what are the aspects of, or the results of hydrogen peroxide, or H2O2 being an oxidizing agent? It will generate free radical species. And these free radical species will do what? They will damage everything in the cell. What do they damage? They, as I said before, it is highly reactive because it is unstable. So it would result in oxidation of various components of the cell. It would result in oxidation of DNA, oxidation of proteins, oxidation of lipids in the cell membrane, and this would result in damage. So that's why we sometimes call hydrogen peroxide an oxidative biocide, because side, the suffix side, means to kill, like suicide. So biocide means that it would result in killing of the cell if it is present in inappropriate concentrations. Why are we adding this at inappropriate concentrations? Because as we have just explained, you will always find a certain level of H2O2 in the cell. Those molecules of H2O2 that have just been formed by aerobic cellular respiration and that are going or being targeted by catalase enzyme to be converted into H2O and oxygen. The second damage of H2O2 as an oxidizing agent is that if it oxidizes DNA, then it would prevent progression of the cell cycle. It would result in an arrest of the cell cycle. What is the cell cycle? During mitosis, there will be repeated events in the cell that are called the cell cycle. And in order for mitosis or mitotic cell division to occur correctly and without any error, there are checkpoints. Every now and then, the cell stops, and there are certain proteins and enzymes that check whether, for example, DNA structure is okay and correct, if DNA replication occurred, if DNA synthesis and duplication occurred correctly or not, before the cell divides into two and gives daughter cells. So if H2O2 is a biocide that results in oxidation of DNA, so definitely there will be no progress or evolution in DNA synthesis. The cell cycle will stop. Cell division, of course, will stop, and this would result in cell death. But paradoxically, there is something that seems completely contradictory, which is that H2O2 can act as a promoter for cell cycle progression by doing what? H2O2, as we said, is an oxidizing agent. So if we leave it to act, it will oxidize. And it will oxidize specific thiol proteins. Thiol proteins contain sulfur. These thiol proteins stop cell progression. So if we oxidize them, this is beneficial because if they are oxidized, they are inactivated, they do not act anymore, and the cell cycle progression. But this seems very contradictory. So how can we explain this paradox? It all depends, as we said before, on the concentration of H2O2. So in these three functions, A, B, and C, it's all about oxidation. But does oxidation when is oxidation harmful? It is harmful when we have high levels of H2O2. And when is it beneficial? Not only beneficial, it does exactly the opposite when we have lower concentration of H2O2. And by controlling the cell cycle, of course, we will be studying the cell cycle in full detail in Unit 4, we will control growth of the cell or growth of the individual, because if you control cell division, you control growth of the individual, and you might also control cell growth and death through control of the cell cycle. We'll be explaining this in full detail in Unit 4. Again, in low concentration. So starting C, we have beneficial effects. D, controlling cell growth and death, triggering differentiation of undifferentiated newly formed cells, again, when in low concentrations, activating immune cells, and therefore killing microorganisms, either directly. H2O2 can kill microorganisms directly, but can kill microorganisms through activation of immune cells. All this happens when H2O2 is produced in a certain range that is rather lower than the range that causes harmful oxidation. Catalase enzyme, as we said, helps minimize the amount of H2O2. Although this again looks paradoxical, catalase enzyme is an oxidative enzyme, but again, it minimizes the levels of hydrogen peroxide, and therefore this is how it reduces damage to the cell. So if we ever ask you how catalase reduces damage in the cell, by reducing the amount of hydrogen peroxide. And you might be asking yourself, can we please look at the top of the screen to the left? How is catalase enzyme an oxidizing agent and meanwhile minimizing the concentration of H2O2, which is an oxidizing agent? If you look at the difference between H2O2 and the products, which are H2O and oxygen. So here you would, if we take those two oxygen molecules, they're here, H2O2, yes. So we have here 2H2O2 gives rise to 2 molecules of water plus 1 molecule of oxygen. Let's count the number of oxygen atoms. 2 multiplied by 2, 4. So we have 2 here and we have 2 here. So actually what it did, if we assume that this O2 is the same, so you have here 2 hydrogen molecules to which we added 2 oxygen atoms, and adding oxygen is oxidation. So H2O2 is a byproduct of aerobic cellular respiration. As you can see here, this is hydrogen peroxide that is produced inside the mitochondrion, and that is converted into water. And of course, H2O2 has been produced during aerobic cellular respiration in the presence of oxygen. But we will see during our study of the chapter or the unit on aerobic cellular respiration how this is related to aerobic cellular respiration. We might explain it in detail, but you will never be asked about that complicated pathway because it will need a lot of details to be understood that are unnecessary to your AP final official test. And now we come to the ribosomes. As we said before, the ribosomes should be present in all living cells, whether prokaryotic cells or eukaryotic cells, because the ribosomes are the sites of protein synthesis. And although ribosomes are assembled in the nucleolus, and we will always use the term assembled, as we said in the first video, yet they act in the cytosol. So proteins are synthesized in the cytosol, and therefore the ribosomes are assembled in the nucleolus and then leave the nucleolus to reach the nucleoplasm, and then leave the nucleus through the nuclear pores to reach the cytosol. What are they formed of? They are formed of ribosomal polypeptides and ribosomal RNA. And we said before that the ribosomal RNA is formed on specific ribosomal RNA genes that are present on the DNA molecule, similarly to the other type of RNA, that is transfer RNA. The only RNA, so we have ribosomal RNA genes that determine the formation of ribosomal RNA molecules, of course, by a process of transcription, as we will understand later on. But we also have transfer RNA genes. The only RNA that does not have genes is the messenger RNA, because I can't say it does not have genes, but every messenger RNA is built up complementary to a sequence on DNA. So every messenger RNA represents the complementary of a particular gene on DNA. But for the ribosomal RNAs, there are specific ribosomal RNA genes for all ribosomal RNA that enters in the formation of the ribosome, and similarly transfer RNA. If we don't understand this very well now, we will be understanding it more and more upon studying the unit on molecular biology. The ribosome, as we said before, is formed of a large subunit, which we usually call the 60S subunit, and a smaller one, the 40S subunit. And the ribosomes can stay free in the cytosol, or they can be attached to another organelles that looks like channels traversing the cytosol, called the endoplasmic reticulum. And if this tubular endoplasmic reticulum, if those channels that are called, or tunnels that are called the endoplasmic reticulum carry ribosomes on top of them, they are called the rough, or they become the rough endoplasmic reticulum. If not, then they would be the smooth endoplasmic reticulum. As we said before, the ribosomes are the site of protein synthesis, meaning that the messenger RNA that is the deputy of DNA in the cell will leave the nucleus. It comes from the nucleus, so it is one of the structures that leave the nuclear pores, and it goes to the cytosol. And it, as you see, it during the process of translation, meaning during the process when messenger RNA is converted or translated into polypeptides or a sequence of amino acids, it lies between the large and the small portion of the ribosomes. Those ribosomes that will be floating in the cells will produce proteins, but only those proteins that will stay in the cell. Ribosomes, on the contrary, that are on top of the endoplasmic reticulum will also produce molecules of proteins, but since these molecules of proteins are synthesized inside tubular channels, then this would be a good opportunity for them to travel through the cytosol to the cell membrane and leave the cell altogether. So both floating and attached ribosomes will synthesize proteins, but the floating one synthesize proteins for the cell, the other one the attached one synthesize proteins to be exported outside the cell. So what is the endoplasmic reticulum? As you see here, under the electron microscope, this is the nucleus, and the nucleus is connected to a series of interconnected tubular channels that are surrounded by a membrane that looks like the cell membrane. Some of these tubular interconnected channels have ribosomes. These form part of the rough endoplasmic reticulum, and the others do not, and they form part of the smooth endoplasmic reticulum. There is a third type of tubular channels that we call the sarcoplasmic reticulum. And when we say sarcoplasm, we mean the cytoplasm inside the muscle. So sarco here refers to the muscle, and here is the sarcoplasmic reticulum, those yellowish tubular channels, and we will be studying them upon studying the structure of the muscles. So this endoplasmic reticulum, we call it endoplasmic, of course, because it is embedded inside the cytoplasm. So it is a system of membranous channels and flattened sacs, sometimes flattened sacs because, as you saw, they might look like this. In between the membranous channels, you might find like flattened sacs. This part looks like a flattened sac. If this is a sac that is surrounded by a membrane and then you flatten it down or you push it down, it will look like this. So it looks like flattened sacs and channels that traverse the cytoplasm. As we said, the rough endoplasmic reticulum, of course, is a site for protein synthesis since it carries ribosomes on top of it, but the smooth endoplasmic reticulum that does not have ribosomes will not be the site of synthesis of proteins, but it might help in transport of these proteins from the rough part, the smooth endoplasmic reticulum, might transport those peptide or protein molecules from the rough endoplasmic reticulum, where we have ribosomes and where they have been synthesized to the Golgi apparatus or to elsewhere in the cell. But the smooth endoplasmic reticulum also has other functions like synthesizing lipids, like steroid hormones, for example, connecting the rough endoplasmic reticulum to the Golgi apparatus, as I've just displayed on the screen. It also helps in detoxification. So organs that carry out detoxification or cells that carry out detoxification, like liver cells, will be rich in smooth endoplasmic reticulum. So if you are asked, as I told you before, about a cell that is rich in mitochondria and therefore be an active cell, and also rich in smooth endoplasmic reticulum, so it's not only active, but it's also rich in the smooth endoplasmic reticulum that detoxifies, and therefore it might be a liver cell. In case this third or fourth function, that is this fourth function, that is storage of calcium ions to facilitate muscle contraction, because you will be learning that calcium is important for muscular contraction. For a certain level of calcium ions is important for normal muscular contraction. So in this fourth point, we are, of course, talking about the sarcoplasmic reticulum. It is important for not only calcium storage, but calcium release. Calcium storage when calcium is not needed for muscular contraction. Calcium release when we are now in need of calcium ions for muscle contraction to occur. The Golgi apparatus lies near the nucleus, and the Golgi apparatus consists, just like some areas of the endoplasmic reticulum, it consists of flattened membranous sacs. But these flattened membranous sacs are not only flattened on their own, but they are also parallel to each other and compressed against each other or stacked next each other. The main function of the Golgi apparatus is to receive proteins. So the main function is to receive proteins and modify these proteins, make changes in those proteins, and then package them, okay? And then secrete them to other parts of the body, or to other parts of the cell. So if this is the rough endoplasmic reticulum, as we said, the rough endoplasmic reticulum is always associated with ribosomes for protein synthesis. And then these proteins are transported via the smooth endoplasmic reticulum. Parts of the membrane of the smooth endoplasmic reticulum form vesicles. Vesicles that separate from the smooth endoplasmic reticulum, carrying with them the protein. So the protein is transported here, and then there is a part of the membrane of the endoplasmic reticulum that is punched out, that is punched out, and we now have a vesicle. This vesicle is very near to separation from the smooth endoplasmic reticulum, and now it becomes a separate vesicle that floats in the cytosol until it reaches this cis surface of Golgi apparatus. Golgi apparatus has these flattened sacs have a cis surface that is rather convex, that receives. So the cis surface is the receiving surface, okay? And then these received proteins inside vesicles will be modified. Modified how? By adding a functional group, or by removing a functional group, or by carrying out further folding, for example. And then they will leave again via vesicles through the surface, the other surface, or the other face of Golgi apparatus, okay? These vesicles usually leave the cell. These vesicles are usually secreted by the cell, because these vesicles will reach the cell membrane of the cell, and then they will fuse with the cell membrane of the cell, and then they will rupture and release the protein outside. So we can understand now why Golgi apparatus is abundant in secretory cells. All glandular cells that have secretions, or even if it's just a secretory cell without being arranged in the form of a gland, it is abundant in secretory cells. This is the first important thing to know. But the second important thing to highlight here is another concept that comes in more sophisticated questions, which is the membrane traffic into and out Golgi apparatus and the membrane balance. And you should differentiate between what we call the membrane traffic and the membrane balance. Membrane traffic is the traffic of the membranous vesicles inside the cytosol. So when we talk about membrane traffic, we talk about the transport of vesicles and membranous structures in the cytosol. But this membranous traffic is always associated with either a positive membrane balance or a negative membrane balance. So the positive and negative membrane balance are associations to the membrane traffic. A positive membrane balance occurs when vesicles fuse with Golgi apparatus, because if you have here one of the sacs of Golgi apparatus, and you have a vesicle here that comes to fuse with Golgi apparatus in order to pour down its content of proteins, then now this membranous sac of Golgi apparatus is larger in surface area. So this is a positive membrane balance. But on the other hand, if this is the membranous sac of Golgi apparatus, and now that this protein has been modified and packaged in a vesicle inside Golgi apparatus, it was once there, by the way, and now it has been modified and it's leaving via a vesicle. So this vesicle will be punched out, and then healing will occur here. So this is a negative balance because you removed part of the membrane of Golgi apparatus. So this is a higher complexity question: membrane traffic, positive membrane balance, negative membrane balance, and please retain this in your mind. Next, we're going to talk about the lysosomes. Lysosomes are present, as we have seen, in most cells. Lysosomes are present in most animal cells, or in all animal cells actually, but they are generally not found in plant cells. Of course, there are exceptions, but usually they are present in animal cells. And lysosomes are membranous vesicles that contain digestive enzymes. And as you know from your previous study of biology, that digestion or breakdown of complex food molecules occurs through hydrolysis. And hydrolysis is the breakdown of substances by the addition of one or more molecules of water. And lysosomes are amongst the organelles that are surrounded by a single membrane. Of course, you know that digestion occurs inside the human body, in the digestive system, in the stomach, in the small intestines, but inside the cell, lysosomes carry out the process of digestion. So it's digestion in every cell, and that's why we call it intracellular digestion. So be very, very cautious when we ask you about the difference between intracellular digestion and extracellular digestion. Intracellular digestion is the digestion that occurs inside the cytosol of the cell or inside the cell, bound by the cell membrane, by the limits of the cell membrane. But extracellular digestion is the digestion that occurs in the lumen or the cavities of the organs of digestion, like the stomach and the small intestine. What's the importance of having this lysosome or this bag of digestive, and be careful, be careful, because lysosome is not lysozyme. Lysozyme is a digestive enzyme. Lysozyme that digests, for example, cell walls of bacteria. And it is present, as you know, in saliva, in sweat, and in bodily secretions in general. But the lysosome is an organelle. So why is it important? Because the lysosome secretes digestive enzymes or hydrolytic enzymes, and these enzymes help intracellular digestion. What's the importance of intracellular digestion? Intracellular digestion helps the cell to renew itself. So be very careful, because there are several mechanisms of renewal of the cell. One of the mechanisms of renewal of the cell is breaking down the cell and recycling the cell. Because when you break down the cell, you have products of digestion of worn out organelles, for example. These products of digestion are not thrown out of the cell. They are recycled to form newer molecules. And we call this autophagy. Auto means self and phage means eat. So it seems as if the cell is eating, or not actually eating, but breaking down itself and taking the products of breakdown, recycling them again to form newer molecules. And therefore, lysosomes are an essential part of a very famous process that occurs in the cell, which is called apoptosis. What is apoptosis? Apoptosis is programmed cell death. Apoptosis is programmed cell death. Why do we say programmed cell death? Because this autophagy that occurs in the process of apoptosis occurs in a very neat and orderly manner. It is very neat, it is orderly, it is as if you were packaging products of breakdown of the cell in small garbage sacks to reuse them later on. But this is very different from another process. So apoptosis is very different from another process that is called necrosis, and that is not programmed, that is not neat, that is not orderly, and we will see the difference later on. Usually we ask you a lot about apoptosis, but we rarely ask you about necrosis because it's not part of the curriculum. And of course, lysosomes act by hydrolyzing the substances we want to break down. So lysosomes are full of hydrolytic enzymes that are essentially proteolytic enzymes to break down or hydrolyze protein components of the cells. And these proteolytic enzymes are called caspases, and they trigger cell death by breaking down or cleavage means break down specific proteins in the nucleus, specific proteins in the cell to get rid of the cells that are unwanted or that have passed a certain number of cell cycles. Because after the cell passes a certain number of divisions, it has to stop because it might commit an error during division, so it had better stop cell division. So we will see in detail or explain in detail apoptosis and see how the caspases act and what cells undergo apoptosis and when. Here we have several examples of the action of lysosomes. So a lysosome, for example, may fuse with a food vacuole. As you see here, this is one lysosome, this is another lysosome, and they both fuse with the membrane of the food vacuole. The food vacuole is a small vacuole that stores the surplus food material, for example, in amoeba or in unicellular organisms. So it might be important in digestion of food, but mainly in unicellular organisms, because multicellular organisms do not store food in food vacuoles. But as you can see here, those, by the way, 1 and 2, are empty lysosomes. Why are they empty lysosomes? Because they have emptied their content of enzymes into the organelle. But these are 1 and 2 before having emptied their enzymes. What are they doing here? They are emptying their enzymes into a mitochondria, but of course it's not a healthy mitochondrion. It is a mitochondrion that the body found out, or the cell found out, that it's high time for it to be broken down. Are we going to throw the components that result from breakdown? No, we are going to reuse all the molecules and the atoms to form a new mitochondrion or a new something else. The third function is to digest the cell as a whole. So it might be important to digest a food vacuole. It might be important to digest a mitochondrion. Of course, this will occur only in aerobic organisms, because only those organisms that respire aerobically that have mitochondria, but it might also be responsible for digesting of the whole cell. And of course, this occurs in the process of apoptosis. Apoptosis, here it is. So apoptosis is called cellular suicide. Why is it called cellular suicide? Because it is suicide. When we talk about suicide, we talk about death that a person does to oneself, or that a person does to themselves. So suicide means here that the cell kills itself. Because yes, this is very true, because the cell dies, but sometimes it is the cell that calls for death, that calls for the enzymes of suicide, that calls the lysozymes to come and start killing the cell in a neat and well-organized manner. So it is an intracellular program. It is a program because it is highly organized, accurate, occurring in definite steps. So that's why we say that it is a program, and that's why we say that it is a programmed cell death. And it is intracellular because it all occurs inside the cell. It does not occur in the cavity, for example, of the stomach. We do not take the apoptotic cells or the cells that need apoptosis, go into the cavity of the stomach and suicide there, or commit suicide there. No. This is inside the cell, and it occurs through a series of molecular steps, simple biochemical reactions, and it's very astonishing that the cell uses its own hydrolytic enzymes to kill itself. And these enzymes, as you know, already are called the caspases. So the caspases are the enzymes that are present in the lysosomes and that help in killing the cell or digesting the components of the cell. So the caspases, first thing to know is that they come from the lysosomes and that they are hydrolytic enzymes, and don't mix them with the catalase enzyme, which is present in the peroxisomes, and that is an oxidizing enzyme. What's the importance of apoptosis? As we said before, if we have an abnormal cell, like a cancer cell, for example, or damaged cells, so if it's obvious that the cell is damaged or abnormal, this would help getting rid of that cell. Also, cells that are not yet apparently abnormal, but that have a mutated DNA. Of course, if we leave that mutated DNA, this means that this mutated DNA is an abnormal one. It will be converted or transcribed into, this is the verb we use for using DNA to form RNA. So it would be transcribed into an abnormal messenger RNA. Again, this is transcription. And the abnormal messenger RNA will be translated into an abnormal protein. And an abnormal protein can be an abnormal enzyme or an abnormal hormone. Okay, so we cannot leave a mutated DNA. So if mutation in the DNA is detected early, apoptotic processes must start inside the cell. You might be asking yourself, so how come that we have cancer cells in the body if the body is that alert and that all mutated abnormal cells are killed on the spot and apoptosis happens in these cells? Of course, the simple answer is that if you find a cancer cell in front of you under the microscope, so you have to understand that this cell have bypassed this process of apoptosis. Cancer cells do not respond to apoptosis. Cancer cells have certain abnormal mutated proteins that unfortunately block the process of apoptosis. So this is a very high paradox because cancer cells are the cells that need apoptosis the most. However, they are the cells that block the process of apoptosis. Why do they block the process of apoptosis? Because being mutated. So this is important to understand. Cancer cells have abnormal DNA, as you know. Mutated DNA is abnormal DNA, so they will finally produce abnormal proteins. And these abnormal proteins will block apoptosis and they will allow the cell to proceed in the cell cycle. So if this is blocked, if apoptosis, so what we block is apoptosis. Blocking apoptosis means what? That the cell will not die. That cell division will continue. And if cell division continues, you will obtain daughter cells that are also cancerous. Sometimes we don't have damaged cells, as I told you before, neither abnormal cells, nor mutated cells, nor cancer cells, but they're just old cells whose lifespan has come to an end. Why do we get rid of old cells? Because as I've just told you, if we leave them to divide, they might give you mutation. The errors are more liable to occur in old cells. So errors are more liable to occur, in these cells. Also, if a cell is virus infected, because if the cell is virus infected, what does the virus do when it enters a cell? Usually, when a virus enters a cell, it breaks down the host DNA, the host cell DNA. So you actually have breaks down host DNA. And that's one of, and therefore the cell will finally lyse. And if lysis and rupture occurs, then we need to get rid, the cell is already gone. And maybe that this broken down host DNA is a trigger for apoptosis to occur. So in human beings, the cell does not wait until lysis occur as it occurs, for example, in the lytic phase of bacteriophage virus replication, but the cell itself calls for apoptosis. Sometimes we have immune cells that are autoreactive. Immune cells are not here to react against our own proteins or our own human body, but immune cells are here to protect us from foreign bodies. So if the body upon early detection of autoreactive cells that react against self proteins, then this is considered dangerous because this might result into an autoimmune disease. So self-reactive immune cells are harmful and they should undergo apoptosis or cellular suicide. But apoptosis can also occur during the normal embryonic development, because you know that you start as a single cell, which is the zygote, a single diploid cell that has the full number of chromosomes, which is the zygote, and then you start dividing by mitosis until you form a solid mass of cells. But later on, we need to have cavities, we need to have spaces between the fingers. So we can't stay like this as a solid mass of cells. In order to have cavities or in order to have spaces between the fingers, then these cells should undergo apoptosis. And if apoptosis does not occur, so you might see, for example, that some people have a fused little and ring finger just because apoptosis did not occur correctly. So the separation of fingers and toes during the development of the human embryo is because of apoptosis. The tadpole, which is the young frog, originally has a tail, but upon metamorphosis of the frog, meta means after, and morphosis comes from morphology or shape. So metamorphosis means change in shape during development. So during this metamorphosis that occurs during development of the tadpole, the frog loses its tail. And this is because of apoptosis, because if this is the tadpole and this is the hind part of the tadpole, if apoptosis or death of cells occurs here, then they will certainly be a separation between the tadpole and the rest of the tail. Also, these superficial cells, when you, for example, on the seaside, there are superficial cells of the skin that have been exposed to ultraviolet rays. And as you know, that from 10 o'clock in the morning to 3 o'clock in the afternoon, the ultraviolet rays are extremely harmful, and they can, with their high energy, ionize your DNA, but only in the superficial cells of the skin. So those superficial cells of the skin that have been mutated will undergo apoptosis. Lastly, in letter D, apoptosis can occur in plant cells. Yes, if specific cells or specific areas of the stem of the cell or of the stalks of the flowers have been attacked by a fungal or viral or bacterial infection. So these cells will undergo suicide, intracellular suicide, or apoptosis in order to separate the diseased organ and therefore prevent spread of infection to the rest of the plant. So there are multiple uses for apoptosis, and in the next video, we will be talking about the intrinsic and extrinsic pathways of apoptosis, which are a bit less important than the main core of apoptosis. So you will read them to understand more. You read this slide to understand more, but you are rarely asked about the intrinsic and extrinsic pathways of apoptosis. So see you, inshallah, on the next video.